I'm gonna try not to fall out of my chair today. No promises, but you know. Okay, so Claudia, did you need a packet of notes?
Yeah, I was gonna take notes on my phone. Oh. You get correct. Oh, no, I can figure out the kind of things you're asking. Yes, my asthma intern take me out when we have school.
Okay, so this is an assignment. There are no points for it. It's just, we'll go over the answers, so, try and find time to do them. Just so that we can discuss them when we go over them. Okay, so we are going to pick up where we left off, which we finished glycolysis and fermentation. All right, so we are going now to go into pyruvate oxidation and the Krebs cycle. And then if we have time, we'll do the last two steps, the electron transport and oxidative phosphorylation. But depending on time, I'd rather make sure you guys are comfortable with the material because we, can always adjust our schedule. All right, so again, this is glycolysis. We did this in the last lecture in a very reduced form. So this is just looking at glucose and how it changes with every step. You do not have to memorize this. This is just to, kind of look at, and it lists, the 10 different enzymes that are used, to again, just keep doing these tiny little changes until we end up with 2 pyruvates. So again, it seems silly to just make a quick rearrangement, but then it fits the next enzyme. So all of these are necessary for us to take glucose and then eventually get energy out of it. So before we go on, any questions about glycolysis, if you had a chance to review the notes? And everyone's good with where glycolysis takes place? Where would you say?
In the
Not in the mitochondria, no.
It's like in the outside, like it's in between the wall and like the new.
Cytoplasm, which has cytosol in it. Okay. So glycolysis is the only one that takes place in the cytoplasm. The rest are going to move into the mitochondria. Okay. Cytoplasm, cytoplasm. All right. All right, we're not going to do that. All right, so usually I bring in these model kits and you build a glucose molecule and then you build a bunch of 6 little oxygen gases, just to, kind of go through what happens. And then you're asked to basically tear it apart and see how many carbon dioxide you can make and waters. So that's all you need as 1 glucose molecule and 6 oxygen gases. You rearrange it, you don't add anything else to it, and you end up with 6 CO2s and 6 waters. Okay, so that's what our body does for us. And every time it breaks a bond, it releases energy. All right. So the handout I gave you, these discussion questions, so there's 9 discussion questions for you to think about. So again, you don't have to turn them in, but this will really kind of help you after this lecture, see where you might, have questions. All right, so take a look at those. All right, so ATP. So this is the universal energy for all living things. There are other forms of energy, like GTP, but ATP is the most abundant and the most used. So we do produce a little bit of ATP in glycolysis. Does anyone remember the net yield without looking it up? 2 is the net. So it does actually make 4, but we spent 2 to get it started. So the net 2 ATP. Okay. And we do in glycolysis, the way we make the ATP is through substrate level phosphorylation. So again, there's a substrate that has a phosphate on it. An enzyme comes, a kinase comes, removes that phosphate and sticks it on ADP, and it's magically ATP. Okay, that's all that is. And that's what this cartoon is kind of showing. So you have this kinase, you've got a substrate, you tear off the phosphate and stick it on ADP. Now you have ATP. All right, so nothing super difficult with substrate level. All right. So we're still, when we go into pyruvate oxidation, and mainly the Krebs, we're still going to be with substrate level phosphorylation. We're not going to do oxidative phosphorylation until the last step. Okay. Now I don't know if I click on this video, if it's going to work, so I'm going to try it.
We have three molecules in each of them.
You can hear it.
This is cellular.
Okay.
This is where the cells get almost all of the.
All right, so that's a 4-minute video. It is a good video, but I guess I will just leave that up to you guys to watch.
It's still not working.
It's still, I could not get it. I thought I had connected it and I was excited because I thought I could figure it out, but still no go. All right, so I'm going to have to talk to the equipment room and maybe someone can help me out. Okay. All right, and again, there's other videos on YouTube that have lots of explanation for how ATP is made, both ways, substrate level as well as oxidative phosphorylation. All right, so we're done with glycolysis. So we have 2 pyruvate molecules. So we took that glucose and we split it into 2 pyruvates. Now we have to adapt that pyruvate before it can actually start the Krebs cycle. So that's called pyruvate oxidation. So what does it mean to oxidize something in simple terms? Does it gain or lose a hydrogen? Loses a hydrogen. So when it oxidizes, a hydrogen is torn off. Okay. And we are going to convert this 3-carbon pyruvate into a 2-carbon molecule. So we're going to tear a carbon off and it's going to leave a CO2. Then once that happens, then it's able to begin the Krebs cycle. And then there are other ways. At the very end, we'll quickly talk about microbodies again. So they are able to take, fats and, convert them into intermediaries that can then start the Krebs cycle, in a specific location. So we always focus on Krebs starting with glucose at the top, working its way around all the way, but other things can enter at different parts. That's where the microbodies are really helpful in kind of getting a fat ready and shoving it into the Krebs. Again, kind of for the same purpose, is to make some energy from it. My child doesn't have school again tomorrow. They have not had school all week.
You can miss 3 days.
I know. You got to miss high school, right? Jeez. Okay. So location of pyruvate oxidation. So pyruvate moves into the matrix of the mitochondria. Okay, this is where a CO2 will get ripped off by an enzyme. And then now it's just a 2-carbon molecule. So we call that acetyl. Okay, that just means there's 2 carbons there. ACETYL. Yep, a carbon gets removed and it leaves as carbon dioxide.
Wait, so carbon gets removed and it leaves still as carbon?
As CO2, as a gas. Yep. Yeah, so you take your 3-carbon molecule pyruvate and you're going to remove one of those carbons. That carbon leaves as CO2. So every time you breathe out, one of those could be from pyruvate oxidation. Yep, there's 2 carbons left, which means it's an acetyl group. Any electrons that are removed in pyruvate oxidation, meaning hydrogens, NAD plus is going to pick them up. So it's going to go from its oxidized state of NAD plus, it's going to get reduced by accepting that hydrogen, and it'll be NADH. Good so far? Okay, one more thing. So with our 2 carbons that are left, there is an enzyme that is going to attach to that acetyl group, and it's called coenzyme A. So from this point on, they'll continue to call it acetyl-CoA. Okay, that's the 2 carbons from glucose, and then a coenzyme A molecule.
Acetyl CoA, that's the 2 carbons that are left, right?
With a coenzyme A added to it. Yep. So the acetyl is still there. So we've got 2 carbons from glucose, and then a coenzyme A attaches to it. Now it can officially start. This is the starting molecule of the Krebs cycle. Acetyl-CoA. Mm-hmm. Yep. I've got a slide coming up for it. Okay, so
Is it the thing that attaches to the acetyl group is an enzyme?
it's coenzyme A. So they just shorten it as CoA. So as So that is how the coenzyme A attaches? No. Mm-mm. So the recap of pyruvate oxidation, starting with the pyruvate molecule, and remember glucose, there would be 2 pyruvates, so everything happens twice. We remove a carbon and it leaves as CO2. So 1 molecule of CO2 per pyruvate or 2 per glucose is going to be produced from pyruvate oxidation. When we're going to tear 1 hydrogen off per pyruvate, 2 per glucose, and we're going to have, an acetyl group that's left that's going to combine with coenzyme A per pyruvate. All right, so this acetyl-CoA now will be able to start the Krebs cycle. All right, and then this is just, I don't find these little equations super helpful, but this is just showing you start with pyruvate and there's a coenzyme A floating around and an oxidized NAD. For oxidation at the end, you'll have acetyl-CoA, a reduced NADH, another hydrogen, and carbon dioxide. Okay, so pyruvate oxidation happens right after glycolysis, but right before Krebs.
You'd be good again, please.
Pyruvate oxidation is right after glycolysis, right before Krebs. So it's getting that pyruvate ready to start the Krebs cycle. And the reason we keep track of it is because we produced an NADH. So again, that's one of those things that we're going to count up. And we lost a carbon. So no energy was produced through pyruvate oxidation.
It starts after oxidation?
It's pyruvate oxidation starts after glycolysis. Okay, this is the same slide, just you can actually see it. But it's important to note. Okay, so here's our pyruvate oxidation that we just went over. But again, no ATP is made. We did produce per glucose 2 NADHs, but we did make a little bit of ATP in glycolysis. We'll make a little bit from Krebs, and we make the most in our last step. Okay. All right. Now, for the citric acid cycle, same thing as Krebs cycle, same thing as TCA cycle. I don't know why there's so many names for it. What we're going to do, so we've got our acetyl-CoA that's going to start this. So the acetyl part, those were the 2 original carbons from glucose, and there's going to be 2 of those, right? There's going to be 2 acetyl-CoAs because there were 2 pyruvates. All right. So Krebs cycle will take those 2 acetyl groups all the way back from glucose and completely oxidize them to CO2. So we get rid of all the carbon from glucose, and then we breathe it out as we exhale.
The Krebs cycle will take the acetyl back to oxidation.
It's going to finish oxidizing those carbons and then just release it as CO2. And then electrons / hydrogens, as they get removed, they are going to be accepted by the NAD plus or FAD. So these 2 are just coenzymes that are going to help shuttle the electrons to the last steps. Okay, and the NAD plus, I think I told you last time, it was we can make NAD plus, but mostly we get it from our diet. It's niacin. FAD, we do not make. We can only get that from our diet, and that's vitamin B2. All right. So in the Krebs cycle, the ATP that we make, we're going to make through substrate level phosphorylation. The boil down at the end, we are going to get rid of the acetyl completely. They leave as 2 CO2s. We're only going to make per pyruvate 1 molecule of ATP. Per pyruvate, we're going to make 3 we're going to reduce and make 3 NADHs, and we're going to make 1 FADH2. Okay, this is all happening in the matrix, the innermost part of the mitochondria. So test question wise, I could ask you how many ATPs are made for each pyruvate that enters, after glycolysis. There would only be 1, but I could also ask you how many molecules of ATP per glucose, and that would be 2. Okay, so this is what the Krebs cycle looks like. So we start here with acetyl-CoA, so it's just 2 carbons, that's the acetyl part, and then coenzyme A is attached to it. pretty quickly, it needs to attach that coenzyme A to actually have this citrate synthase actually attached to it and convert it to citrate, but the coenzyme A leaves really quickly. So it gets it started, then another enzyme fits it, and it's able to then start working on breaking down and getting rid of those 2 carbons from glucose. The coenzyme A immediately goes back and finds another acetyl group to attach to. So this citrate molecule is the first intermediate, and then another enzyme will convert it to isocitrate. So again, it's slight little changes, but again, they're important. And then after the second intermediary, this is where we get our first NADH made. So we tear off a hydrogen. We also remove one of the original carbons. So we produce our first molecule of CO2. So now we only have one original carbon left. Yeah. Oh, I am. Okay. Yes, I was using my mouse. Okay, so we start here. So here's our acetyl-CoA. You can see immediately the coenzyme A, once it gets it in there and this enzyme, citrate synthase, fits it, coenzyme A just gets knocked off. So then we have our first intermediate. Then the next enzyme will do a slight conversion into isocitrate. Then we'll have NAD come and pick up, tear off a hydrogen from here. And then a carbon also leaves. So one of our carbons leaves as CO2. So this carbon right here.
The whole thing you're pointing to, in all the letters and everything, that's the COA, right? Just broken.
No, the coenzyme A was only up here and it already left.
It did, so that's the first thing.
Yep.
Is that a pair or is it the same COA? Like I looked through a little.
Oh, no, this is like it attached to acetyl and then this is it leaving.
Okay, so then. Where did the
Where did all this junk
Yeah.
come from? From this enzyme. It brought in citrate and
So it makes
attached.
sense,
Yeah, and this citrate really doesn't ever leave. It just stays in the cycle to attach to coenzyme A's, acetyl-CoAs.
okay.
Okay, so they get reused constantly. But it's important because you tear off a CO2, you tear off a hydrogen, then you're at your next intermediate with another enzyme. So now we're going to be able to remove the final carbon from that original glucose. Then another hydrogen gets torn off. It looks like CoA comes back for a minute to fit another enzyme. So here is our first molecule of ATP.
Wait, so, wait, never mind, wait, yeah.
So you don't have to really worry too much about all these intermediate steps, like the isocitrate and the alpha ketoglutarate, because they're not really doing anything other than being a substrate to fit the next enzyme. So what you need to focus on are the number of carbon dioxides that leave, the number of NADHs that are produced, as well as how much ATP is produced.
Oh, okay, so stuff on the outside house.
Yes,
Oh,
yes, so don't get freaked out or bogged down with
yeah, because I'm
looking
just
at these giant molecules, because they just stay in the Krebs cycle.
Okay.
Their whole purpose is just to help break down glucose.
And they're in the crevice layer, you said?
Yeah, this is all in the matrix in the Krebs cycle.
Okay, okay.
Okay, here's where we have our first FAD come, pick up 2 hydrogens, and then it leaves. So we end up with, so we had succinyl-CoA, succinate, fumarate. So a little bit of water comes in with another enzyme, and then we have our final hydrogen that's torn off up here. And then we have oxaloacetate that now is just waiting for the next acetyl-CoA.
And how
Okay.
many hydrogens does it remove, like 2 hydrogens or just add?
So it's going to pull off, so we've got 3 NADHs and 1 FADH2. So 5 total hydrogens are removed from our original acetyl-CoA.
keep what we need to focus on? Like,
Yeah, actually, I highlighted them. So
Yeah.
definitely focus on for an entire spin of the Krebs cycle. This is one pyruvate. Eventually, get acetyl-CoA. You only get one molecule of ATP. Then the last two things you need to also count up. So here are NADHs in the red circles. So we have 3 of them, and we have only one FADH2. Okay, and you don't even have to worry about, what are the intermediates that it's between, or what is the enzyme that's working on it. Just really the total count per spin or per acetyl-CoA, which means per pyruvate. So 3 NADHs, 1 FADH2.
Reduce means remove, right?
What's that?
Reduce means remove, right?
Reduce means a hydrogen was added to it.
Oh.
I know, it should be reversed. I know, reduce, you would think you were tearing a hydrogen off, but that's oxidized. It's added.
That's what it produces.
Yes. Yep. Okay.
Carbon or hydrogen?
Hydrogen to get reduced, yeah. So Hans Kreb discovered this cycle in 1937, and he got a Nobel Prize for it in 1953. So the amount of work that he had to have done back in those times is pretty amazing.
You did, what did you say?
1937 is when he discovered it or published it, and then he got the Nobel Prize in 1953. And then they renamed it. I don't know why they would do that, but they did.
Is it renamed Trump?
No.
He's renaming it.
True.
Please do food here, like those random things.
A root beer?
Oh, no, a new food here, just like Trump.
Oh, yes, and it's upside down and it's really bizarre. Yes, I have seen that. There's a new food pyramid from the,
South Park already did a thing on that like 10 years ago. So it's good. So.
Ah.
Dementia. All we get are just old pedos as president, don't even know where they are.
Okay, same boil down, each acetyl group, which also means each pyruvate. 2 CO2s leave, 1 ATP, 3 NADH, 1 FADH2. So again, if I'm asking you per glucose, you would double this. Right? There's 2 pyruvates per glucose.
It's not pyruvate.
Pyruvate. What were you asking? Oh, so this what's shown up here is per pyruvate, but you would double it per glucose. All right, so that's the end of lecture 3. So let me check our time. Oh, it's only 6:30. Okay, let's take a look maybe at some of these questions. Do you? Excellent. Okay, I'm going to give you 5 minutes to take a look at these questions, and then we will go over them rather than just have you do it for homework. So 5 minutes, it is 6:32.
These are just beyond little tasks, right?
NADHs, yep, and FADH2s, yep.
Oh, yeah, you just have.
Okay, so if you got through question 6, the 7, 8, 9 are the last part, so you may not have been able to answer those yet. If you put an answer, that's fine. Okay, good.
No. Wait, hold on. Yeah, I probably.
Okay, oops, no, not yet.
Yeah, but they start as that.
Okay, so for the first 6 questions, how about if each of you answer one? So who wants to take question 1? Okay, Gaia. Where in the cell does glycolysis take place? Good, cytoplasm. Okay, how many ATP molecules are needed to start glycolysis? Zach? Yes, it is 2. Number 3, what are the products of glycolysis?
With ACE into NADH.
Yes, good. Number 4, where in the cell does the Krebs cycle take place? Claudia?
Mitochondrial matrix.
Excellent. Number 5, we're not going to do the intermediates. I mean, that's, yeah, no, I used to be much meaner. Yeah, isocitrate, alpha ketoglutarate. Yeah, biology students have to memorize that. It's just like all the structures of the amino acids. It's something we make them do. I don't know why. No, you do not. So number 5, basically, what are the products of the Krebs cycle? Yes, Sam.
Pretty sure it's 3 NADH, 1 FADH, and 1 ATP, is that right?
And that's per turn, per
Yeah.
pyruvate.
Yeah.
Okay, good. Okay. Oh, yeah, you could, yes, 2 CO2 is definitely, add that too. Yep.
Yeah.
Okay, Sonia, you have the last question. What purpose do NADH and FADH2 serve? What's that?
This is the one where I realized Stan. Stan said, I guess, and my I was ready to did trans. I said, I guess they transferred the lots of hydrogens. I was like, yeah.
Yeah, that's exactly, yep. So when we tear these hydrogens off to change it to fit the enzyme, we have to do something with these hydrogens because hydrogen means acidic. So if we don't bind it to something, you're going to be upping that pH really quickly. So it's really safe to shove it in a taxi like NADH or an FADH2, and then it takes it to where the last step is. All right, the last three we will do after we go over electron transport and the rest of aerobic cellular respiration, which is lecture 4. But now, so in this little packet I gave you with the questions, I gave you a beautiful drawing of the mitochondria. So this we're going to need for the last part of this, because even though there's pictures, you know, a lot of these slides are just words, but I want you to be able to see the inner membrane and all of these proteins that are embedded in it. That's where everything is going to happen for the last 2 steps. Yeah. So electron transport and oxidative phosphorylation are the inner membrane that's on here. Oxidative phosphorylation, the last 2 processes of aerobic cellular respiration. Okay. Okay.
Wait a minute, I feel like I know this answer.
Yep. Now my inner membrane space is gigantic. So obviously it would not be that big and far away from the outer membrane, but that's that. All right. And then what we need to talk about is the poster. So in the past, we had enough students to do groups. I don't know if we do this year because we'd only have 3 posters. I know, we'd have 3 posters. So I think we're gonna do individual this year. Now you can do them digitally. You could, you know, go get poster board, you could tape paper together, whatever you want, whatever size you want. But you are going to do aerobic cellular respiration. So that means starting with glycolysis all the way through to the end of oxidative phosphorylation. So I'm going to skip these and I'll show you some samples. Okay. So again, ignore the group part. So you're going to create a poster. We are going to divide you into halves. So when we start this and hang everything up in here, half of you will be standing at your poster. The other 3 will circulate and visit the 3 posters. So every time someone comes up, you have to run them through your poster, as well as me. Then we will switch, and then, you know, you'll each get a turn presenting your poster multiple times. When I come and visit, I'll be asking you questions. Yeah. But it's all just to help, you know, the more time you spend with this and you have to repeat it, teaching something is one of those study skills. If you have to teach it, you learn it differently and you learn it in a deeper way. So you can, this is optional, but students from the past, you know, they picked their favorite organism and then just, you know, basically did a cell, because all of our cells do this. So they just picked a cell to do the aerobic cellular respiration in. So you'll see that on the examples. So you can do this as a flow chart, you know, starting with glycolysis all the way through. All right. And then you guys will also be grading each other. So most students give everybody fives, which is fine. This is peer grading. So you can, you know, if you guys are artists as well. So if artistically you think it's very informative and really well done, or if, you know, you just don't want to be mean, but it's still a good exercise to go through to peer grade each other. So you're going to look at the main ideas and concepts are correct. Also, the relationships between the different phases of cellular respiration are established. The graphics are well done and labeled correctly, which means spelled correctly. There are no spelling errors, punctuation, or grammar errors, and the oral presentation was clear. And the presenter made specific references to their posters, you know, actually, you know, use their poster as a tool to walk you through what they were saying. Okay. So here, I don't remember, I took their names off, but here was a digital one. So they have all, you know, they numbered it. So they kind of went through and then they had just kind of the totals at the bottom. And then they had this, and again, I love kitty cats. So here was a kitty cat zooming in on one of the cat's cells. And again, you have to have all of the processes. So make sure, you know, if you don't put a lot of detail on your poster, you're going to have to say a lot of detail. So keep that in mind. If it's really simple, that just means you have to know it even more, you know, to explain it. And then we just had a, you know, no animal, but just kind of um, uh, a cell, um, a rooster. Okay. And then this one was very artistic, but they had all the parts. Yeah. This one was this one is like textbook ready, kind of like with the typing and all of that. So again, it just depends on your art style, but have fun with it. So let's take a look at, assuming that we don't have any cancellations. Let's see what your schedules. So I originally on the syllabus, so today's the 27th. So we had this scheduled for a week from today. Is that enough time?
Maybe not, just the time to get supplies, it's like,
Okay,
cause I was driving today and
I know, it's horrible.
I literally had to get pushed out of a snowbank by like cops.
Okay,
I can watch people get out, but. I had to literally swerve in the snowbank because some asshole semis like they fell off. And I shouldn't laugh, that's a rough moment, but also like, I just need, for me, it's just, I need to. See if I actually know what I'm gonna talk about. I'm gonna be honest, I'm one of those people, I'm stressed whether it's doing a week or 2 weeks, so for me, it's.
okay.
I need just other words. If y'all.
So we're probably, because we missed a day, I think I'm gonna have to bump exam 1 to the 12th. So let's have our posters on the 10th. So that's 2 weeks from today. Okay? All right. All right. I think I really don't want to. Let me see. Let me see if there's anything I can start. So for lecture 4. Okay, that's not even showing up. Okay, now, well. Okay. The February 10th, so 2 weeks from today.
And if we do it digitally, you want us to bring in a printed out copy, right?
Yes, yep.
Do you have a size preference? That maybe you want it to be?
Nope. I mean, I have had students do it on just regular, but it's a lot to fit.
It's like print it out, like one for each person, like hand out to people individually. I think we have to pay for a color. That's why I was asking, because like, if you're running lower on your printing budget, you can do a small time flat.
Yeah, absolutely, yep. Yep. And you can even tape two pieces of paper together.
I have, well, the other side of thing, broken printer, I was trying to do like the book, so I really glued them together.
Yeah. Yep, so this is not an art class. I mean, I love looking at your artwork, but okay.
So there's like so the poster, what's like the biggest or smallest, like size of?
It's whatever you need to fit all of the steps and all the guides that you need to talk someone through it. Okay?
The DOC will print much smaller than like.
So, you know, we can pick any organism.
Mhm. Or none. You don't even have to pick an organism. So some of those samples were just a cell. So they didn't want to, maybe they didn't have a favorite animal, so they just did a cell. Some had fun with it and they picked, you know, like that digital kitty.
Raise them different symbols.
No, all the same. I mean, there's variations, but yeah.
But you just do the same process.
Mm-hmm. Yep. Okay, the last two steps are really important, but there's a lot to them. So I'm trying to see if I don't even know if I have a good place that I could stop.
Can we just somewhat review the stuff we've already been over?
Yeah, what questions do you guys have?
It's like probably obvious, but we're trying to get to know like if the like in behind it. And
If a what?
it's like. Like if the hydrogen's been added or taken.
Just if it is, okay, let me see if I can find a good slide that. There was one slide that was trying to actually show. No, that one's not a good one. Maybe it was the last class, or like this one right here. Let me go back through them.
Right now, I feel like you don't. You don't.
I think what we started was if you really figure out we don't have like same stuff.
Okay, this one might be best. Okay. All right, so whether or not something is reduced. So again, this is what NAD plus actually looks like. So you can see that, you know, we're focusing on this right here. So there's only one hydrogen, but when it turns into NADH, meaning a hydrogen gets added to it, it gets added in the same location for every NAD plus turning into NADH. So it's a simple conversion of getting rid of this double bond. And then now carbon is going to be able to bond with that hydrogen that was torn off the molecule.
So the NAD plus is not oxidized,
This is
or it is oxidized, and then the NADH
is reduced. Okay.
How well do we need to know all the fermentation stuff?
Other than,
We keep it back.
oh, which to where I was?
Yes, just really.
Yeah.
Okay.
So with fermentation, all you have to know is really the reason why
Okay,
it's doing it.
so you're not like as worried about us knowing the exact like chemical steps?
No.
Okay.
I might refer to lactic acid fermentation or alcoholic fermentation. But other than that, you just have to know that it's to regenerate it back into its oxidized state. Okay. Okay, so I think I am good to end. I really don't want to start the last two and then have to, but for those of you that have not done this in a while, again, try and look at this stuff briefly, especially this chart right here. So this is just showing the proteins that are embedded in that inner membrane. And this is where you get that separation between the matrix and the inner membrane space. So this is going to be a big focus of what we talk about in the next lecture. Okay, because this is both the oxidative phosphorylation and the electron transport chain. So this is an important thing. All right, I think we're ready to wrap it up. Is it?
Like the top rows.
Oh, it is.
Oh, it is.
That's why I was thinking that, so I don't like it, it's not like it's.
Ooh, all right.
The TV's like color setting.
All right, I'm at my drive home. Yeah. Is anyone else driving?
No, not until the clue's gone. I already did my time.
Guys, I miss having cards from the.
Oh,